Hi, I'm Kim Demora. I'm a PhD student at the University of Edinburgh, and for the last year I've been working in Pam Silver's lab at Harvard. So I'm going to talk a little bit about the measurement kit very, very briefly. So this is something that Jason started to make and I helped distribute to the iGEM teams this year, and from some of the feedback that I've heard back, there's some people that have used it. It's how the Harvard team started their iGEM competition this year. The first thing they did was they took the practice promoter set, transformed it, measured it, and actually got results that were approximately those levels. So that's right. It seems to have worked so far. So this is quite a good model for actually distributing standards. We sent out the kit, and it appears that people actually have started to use it. So what I want to talk about is how I've done something similar in yeast. I've taken the ideas that Jason had and I've tried to build on them in yeast. So I'm going to briefly talk about this collection and how I've set it up. So I've got 29 strains, 13 promoters, and the 13 promoters are all with the human ribosome binding site, or COSAC, and the yeast ribosome binding site. I've got 396 well plates with 3 replicates each, so I can measure up to 9 replicates if I feel like it. And there's 2 constructs in each strain. So this is a departure from the way Jason did things, where you've got a GFP and you measure the GFP expression level. What I've done, or rather, I should say my colleague Jake Wintermute, who originally built this collection, is I've got YFP, a YFP protein with the promoter and the either yeast or human ribosome binding sites and the terminator. And that's different in each strain, but I have exactly the same construct in every strain. The alcohol dehydrogenase promoter, which is quite a common promoter in yeast, the same COSAC, the same ribosome binding site, and the same terminator. So the idea behind that is to start off with a normalization across the population. So you have something that you can compare in each strain, and you normalize to that level, and then you can compare your YFP data on the promoter strengths. It's viewed to be constitutive in yeast, yes. So technically, it's induced by glucose, and I've measured it in a bunch of different media conditions. I'm not really going to show that today because the data is extremely messy and very difficult to present in a coherent manner. But what changes when you change the media conditions is the standard deviation. So it's quite a tight distribution when it's measured under induced conditions, so glucose, but the distribution gets more broad when it's measured under different media conditions.
Sorry, can you say, are these 2 constructs for the same strain?
Yeah, exactly. So, sorry, yeah. So 2 constructs in each strain, and I've got a couple of controls, one of which is just the RFP without the YFP, and so on and so forth. Yeah, okay.
So are you measuring things relative to ADH1, or you put in your reference an ADH1 in one, and then your new one in ADH1, and is your ADH a leveling thing, and then relative is the second one?
Yeah, that's right. In theory, we normalize to the RFP. So in theory, the RFP should be, okay, so I'll show the data. So in theory, the RFP should be same across the board. Sort of. It sort of is. This is not particularly good data. I've got some better data since then, but it's a little bit. I haven't had time to process it appropriately to show. So this is what the raw data looks like, and approximately the first half of the promoters there is with the human ribosome binding site, and the second half is with the yeast ribosome binding site. So that's what the RFP expression looks like. It looks like we might have got a double integration in one case where our construct has gone in twice. And I should say all the constructs in yeast are integrated into the genome. They're all integrations. If anybody's familiar with yeast work, we use the Sikorsky vectors. I think we use His and TRIP, if anybody knows what that means. Yeast are pretty good at losing plasmids, so if you want to get any kind of consistent expression level, you need to integrate stuff into the genome. Yes, the RFP is, yeah, they are. The RFP has been integrated into the same locus in each strain, and the YFP is integrated into the same locus in each time, in each strain. And then, okay, so this is the the different promoters. The blue is with is one ribosome binding site, human ribosome binding site, and the yeast is with the red is with the second ribosome binding site. And you can see roughly the differences between there's a laser pointer between the promoter expression levels. So that's there. The alcohol dehydrogenase promoter. There's quite a difference when you change the ribosome binding sites. In general, the yeast ribosome binding site is better, but not in all cases. So what I want to get to is, with this data, I can pretty much start to build something similar to Jason's promoter measurement kit in yeast. So I would propose definitely using the ADH1 promoter as the standard because it has the best distribution. I've got the FACS data to back that up. And then we could select other promoters varying for different expression levels. But that beckons the question, do we really want to do that in terms of standardization? How useful is going to be making another measurement kit for yeast? Is that something that we want to do? And if we do it, are people going to use it? Is it too early to start adopting something like that? Or do we really want to start pushing standardization, like that kind of standardization? That's pretty much where I want to leave stuff. And on that, I'll start the discussion.
So when you measure relative fluorescence, is that all the same OD?
Yeah, it's the same as Jason's method. All the strains are the same OD roughly, and it's all in mid-log phase.
Yeah, that method assumes that the relationship between fluorescence and OD is completely linear, and I've seen some actual non-linearities in that. Yeah, so yeah, I kind of wanted to ask about that. You just take a relative fluorescence with a single OD, but that's not necessarily representative of the expression per cell, like a population kinetics situation. So essentially, I measured the Jason promoters, and they kind of go up. There's looks like there's some lag phase nonlinearity, and so I took the slope of fluorescence versus OD for the linear region, and it was actually it changed the rank order, one of them, as opposed to just measuring a single OD and comparing them.
Yes, the data I showed. So the data that we did, that I was showing there, was all from plate reader time courses. So we were looking at GFP synthesis across, I guess, probably a 40-minute range in the mid-log, and that was pretty. In our experience there, anyway, it was relatively consistent.
Yeah, I got actually really close numbers to what was presented in the measurement kit when I did it that way. But the problem with that is that you're assuming that it's completely linear fluorescence versus OD when it's actually not.
Or maybe the answer is to just do a time course and see what it looks like. I mean, I took all my readings with FACS, so it's a little bit more difficult than on a plate reader.
With the measurement, what we recommend in the instructions is to do 2 time. To do 2 time points or whatever, so people can take a synthesis measurement rather than a single time point measurement.
Well, actually, I did the whole time course with the mental and that's what we used.
Okay, yeah, we could talk through it.
So do you think one of the issues is maturation of GFP?
Yeah, I'm not sure what is the maturation time.
It's very easy.
What's the maturation time for the constructs that we're in the measurement kit?
Yeah, so I think I have to look at it again. I feel like it's less than like 7 to 10 minutes. We didn't measure it directly, but we had that same GFP had been measured previously, so we'll have to put that in pretty good.
And that's quite quick.
The GFP matures pretty quickly to equalize those.
On the previous slide to that, where you had so the RFPs are all different. Those are each 9 replicates, right?
There are 3 replicates.
3 replicates. So
That was the average of 3 replicates.
is that variance real, or is that just technical variance? Do you have some notion of where the error bars would be on that chart?
Unfortunately not. I still need to go back through my data and do a little bit more comprehensive analysis. I've done it a couple of different times on different days.
Do you think it's real? The chart record did state there that they actually come out that differently.
I would say when I took those measurements, the machine wasn't correctly calibrated. Yeah, I think there's technical issues regarding how you set up your machine, how you set up the next machine when you're taking the measurements. Just having done it again, I get much closer data. Yes?
Your measurements are based on ensemble measurements. If you look at distribution of expression in the individual cells.
Yes, so each peak was the average of 3 replicates, and each replicate I measured, I think, 10 or 20,000 cells. So there is a distribution in that, but I was just looking at
how wide is the distribution?
I was just looking at the mean for this example.
Sorry?
I was only looking at the mean for this. I have standard deviation measurements somewhere, but I don't know them off the top of my head.
An order of magnitude or 2 orders of magnitude?
Depends on the media condition, probably a couple of orders of magnitude.
There's any of the E. coli promoters with what you use?
I haven't tested them. I don't think so. But that's maybe something to test. Yeah.
I was just wondering, maybe this is a question for the group in addition to the campsite. Do you think that the field is ready for standards for yeast? How many
promoters
are there in the registry that are yeast specific? And would it be best to make standards before a number of promoters appeared in the registry, or would it be best to wait until those are there? What do you guys think?
I don't think there's enough. I don't think there are enough parts in the registry for people to be able to work on yeast if they're not a specific yeast lab. Like I've got these parts because they come from our lab collections. So I don't know. Do we want to implement the standards before people can actually use them, or do we want to just let people go out and
how many people are working on yeast synthetic biology?
Okay, maybe, in terms of the item competition, for example.
Oh, I see.
Yeah, there's not that people can do it and solely working from the registry, but there's not that much that you can do it. I think there's maybe only 12 or 15 parts that definitely work, and maybe 20 to 25 parts total.
But it will probably become a lot bigger.
There's this stuff all in the registry.
I think the one way to approach it is to just bootstrap with some collection of promoters that you measure ahead of time. So if you measured a set of constitutive promoters that span a pretty big range, and there's 20 of them, that almost solves the constitutive promoter issue, right? That might give the range that people need, and then you're just saying if you go measure some other promoter that responds to some small molecule yeast or whatever, or compressed, it just you could use that to set the reference standards, and maybe people will come along later and use it. I don't think it's crazy to start.
Yeah, maybe having something like that, 20 characterized promoters will be enough for identities to start working with yeast.
Yeah, I think that would be valuable.
Any other questions?
Just bringing back to the points of discussion from earlier, I was wondering if you guys had any ideas about how to encourage teams or groups to implement measurement standards. For example, should there be some incentive in the registry to provide measurement data other than honor?
Well, I feel like the thing though is that the key is to just, if people are going to be measuring already, trying to get them to do it in a way that's standard, right? So it's not necessarily that you have to encourage people to measure a whole bunch of things that weren't going to do it before. I don't know that we necessarily have to have a prize for. Maybe it would be cool to have something for measurement prize, but the key point is if someone's going to go out and measure something anyway, they may as well do it such that their measurement's actually reusable by another person other than themselves, right? Which is sort of the current state of the art, in my opinion.
I think knowing that someone else might find your measurement useful makes you more likely to do the measurement. I agree. There's
The other thing is publications. A couple of us are interested in promoting publication of writing standards for which you have to document some kind of functionality.
The open session.
So what would that be? That you mean like in order to get a part published as this sort of
As a data sheet.
it would need to adhere to some.
It's clearly a stopgap for this electronic publication format, which goes straight to the database. But you know, if you think of the equivalent of electronic cards, where there are data sheets which would at least provide a link to the sequence itself, but also some kind of functional description. And so I've been sharing chats with Adam Arkin as well. So I think there are a couple of journals who are really interested in promoting this, which the conventional side, which does not cater to it.
Right.
And I think there's a real need for that. So it will happen anyway.
Yeah, that would be cool. So basically embed the standard, like embed adherence to particular standards and getting a data sheet published.
That's a pretty strong motivation.
Yeah.
People want stuff on this.
So for data sheet,
Jim, on that point, I think don't we have to agree a set of standards?
Yeah, well, I think that's one of the things that we're talking about is how you would put that together. And I mean, personally, I think that the GenBank sequence format, sequence description, provides a starting point for annotation. Software is already existing to allow compilation of sequences that way. And it's purely composition, so it's not, it doesn't really incorporate functionality. But you can have an XML format which will allow easy print and web publication and submission. But I mean, that's the first vision. Sort of baby step, essentially.
I was going to say, for a data sheet for standardized, for a data sheet for a standardized part, would it be a bunch of different graphs showing different media, growth phase conditions, blah blah blah, or is that completely ridiculous to consider every variable possible?
I think the point here is not to be descriptive, but
Okay.
rather to be inclusive. So if anyone
Yeah.
provides
So whatever you have.
use of functional definition.
Yeah.
If you've got a card, you can demonstrate that it's useful.
Right.
And so you can do that in many different ways. I wouldn't be prescriptive.
Right.
Anyone have any points they want to address while we're all here?
one of your initial questions was what other parts do we want to characterize? Does anyone, is there interest in characterizing terminators?
I don't see toxicity. Toxicity, that's actually really high on time. That's higher than expression level on that list.
What do you mean by toxicity?
Like, say, you always put a note for each thing, so in front of the same p-bad, and you measure its growth rate relative to GFP under p-bad.
Okay, so that would be on the red molecule of
Selection of all that, or whatever else is in category. It's probably less that than more like, how bad is this transcription factor? You know, like things that just are toxic. A lot of transcription
and
factors.
you measure the toxicity simply by measuring growth rate hit?
That would be minimal. I think more, I mean, there are other ways toxicity can manifest itself. That
So you want some sort of standard checkpoints, I guess, on the cell that measure how well its overall system is working, something like a diagnostic, self-diagnostic kit.
more just to know that a coding sequence is toxic and you turn it on ahead of time. It's like how like low, medium, or high bad.
Yeah, how precise does that need to be? Is it going to be this coding sequence kills the cells and they don't grow at all, or this affects the growth rate a little bit, or how
I
do
think
you?
all this stuff, how you interpret these numbers is something that you only find out later. You have to start taking some measurements, and then through experience you start to learn what these numbers what degree quantitatively that process rewrite means something, but definitely just give you data points where at least you know that damage from nutrition, when we work with that, it's really toxic even in a cell or calculated. Knowing how bad it is something very useful for migration.
How else do you measure toxicity beyond growth rate?
So you can get things that don't cause toxicity in terms of growth, but you see other things like some morphology changes. You might see get some transformation efficiency where they're putting it into a cell or putting in another plasmid on top of a cell that has it. So there are all sorts of things that can manifest themselves here. So going beyond just growth rate is probably hard to do everything.
You need quite a comprehensive scope of measurements.
Like growth rate's pretty nice.
Yeah, I
pretty
would
convenient.
say growth rate might be an easy one to start with.
Yeah.
I think something that you touched on earlier as well, which I don't know whether maybe can be an issue in time, as like the circuits that people are designing are becoming more and more complicated and more having more and more variables in there, is a kind of physical load on the cell. And like we're talking about characterizing promoters on potentially high copy plasmid numbers, but what if you start having gene cascades of 2, 3, 4, 5, 6, and all of that, and how do you then build that into all of the variables that we've already talked about in terms of promoter strength, ribosome binding strength, protein load, toxicity, and it only seems to be coming as we progress, and bit by bit, is it an issue, and should we, how do we go about this? Because at the moment, if you're realistic about it, it seems that we're a small but growing community of grad students that have and postdocs that have the time to get in the nitty gritty at this data, but there's so many variables at the moment that we're kind of, and that's obviously why we're looking for standards and nailing ones down. But the feeling is, I get the feeling, especially with some of the practical work I've been doing recently, that the more complicated we make the circuits, even on a really relatively simplistic level, there is still this issue of like how much resource does the cell have, and that can have a huge effect then on your readouts, because you're not necessarily have direct access to just the promoter in that circuit. I don't know what people feel about that.
Yeah, I think that's very important, like knowing how there's system-wide effects and being able to see what influence that has on the measurements you're making. Because a simple thing, if your promoter is reducing all protein production, then the readout you're getting is not due to your promoter, or it's an indirect effect. There might be various other things. So it would be nice if there's some sort of diagnostic kit that tells you, check these parts of the cell. Protein synthesis is working properly, and then other things are working, and then you can say, well, this particular circuit affects something, your amino acid pool.
It's kind of a research effort, but you can imagine expression promoters being turned on, you have production of GFP things, and reporting how much GFP got turned on in that cell.
Which have all causes stress and then you can have another set.
Gotta start somewhere.
Variant play with putting like a switch of GFP on the genome, and then you have that's kind of like a power meter, and then you have your second construct, whatever it codes, and if that influences the expression of your power meter, then you know that you're having some impact on the protein expression of the rest of the cell. It's kind of an interesting way of like detecting.
It's pretty easy to see on the growth rate. You see that, if you turn off, the growth rate goes, and then pick up again.
Yeah.
Very, very nice.
I agree, but that's a.
But what do you do about it?
Exactly. What do you do about it? And when we're testing these things, you don't know how far into that window you've got. You've got X units to distribute between you, so that you use.
You're okay if you're dealing with steady state, I guess, but it really affects the transients.
Well, it also gives us a better feel for where the load, where the biggest hits are from the load. Like what's, is it we're running out of amino acids, like what transcriptional machinery, right? So what is it that gets depleted first? And because I think the growth rate's a great place to start, but then I think there's a whole series of things that you'd want to figure out where it's coming from.
Is it possible to create a strain that's better, or are the strains already optimal for that? Just shows the E. coli kind of grows fast as it can anyway.
It'd be nice to have a chassis standard that you could use a model for its ribosomal activity, the nucleotides, all that stuff.
Yeah, I actually have a question. Why did you choose that?
Just ease of use, just in that we figured that's what people would have available to them. But we tested a second strain. That was that one data there that you saw where there's a pretty big difference. But top 10's kind of annoying because it has certain media requirements. But yeah. I mean, one type of cell that might be interesting would be one that where the growth rate isn't really tightly tied to like where a little bit of load wouldn't lead to an immediate growth hit, like where there is some buffering, basically, you know.
I'm wondering if it's possible to beat that decoder. It's already
Well, I think
top performance.
you do the reverse and put it all in the genome, right? I mean, at some point you need to be building this on the genome, and probably the point is like increasing that. And so with plasmid encoded stuff, it's often going to cause, especially high copy, it's going to cause metabolic load almost inevitably. Like even if you express at a low level, it's going to cause metabolic load. So as soon as you go to the genome, you can start to probably more operate within that buffer.
But would you need something more sensitive if it's on the genome? Because then you'd be getting considerably lower levels of expression. So.
Yeah, I mean, I think GFP can easily read off the genome.
I mean, I think the key is to distinguish between the system that's operating and like a reporter, right? So like you can, I mean, who cares if your whole system, I mean, barring like stochastic issues and stuff, I mean, you might just be happy to be running your system way lower. And then, yeah, reporters might still need to be high copy.
Anyone that's interested in making a system for easily redefining bio-based stuff on the genome. Been on the hit list for a while.
Yeah.
Happy to talk about it.
Much needed.
Anything like that with viruses? Yeah. We're working on it. Is it report?
Okay. You can do that in yeast because they not supposed to be vectors. 3 of the 4 are fiber.
Yeah, I mean, I think yeast is conventional, but
There is method off. I mean, there's transposons, phage integration, alpha integration, and then like the red stuff.
yeah, no, I'm not saying it's impossible. I'm just saying, yeah, you just want to make it like a very dead process. Yeah, exactly. I mean, I don't think it's not a
A lot of research.
yeah, it's not a massive research project. It's let's make this tool to make